Today's video is going to be a continuation from last week, in which I talked about mutations and taking that concept into the evolution of phage genomes. Evolution of phage genomes can actually serve as a model for all evolution, because all the same processes that occur in the phage can be found in higher organisms, including ourselves, and essentially all the same processes that we have that lead to evolution of higher organisms can be found in the phage. So all genomes do evolve over time. That's a given. There's always going to be change going on. And this happens at the level of DNA. DNA is an unstable molecule that leads to this constant change and providing the cell or the organism in general with a reservoir of new material that it can be used to build or react to the environment. Changes can be small or large, and we talked about that last time. And any change, though, to be involved in evolution, it must be retained. And that means it must not be repaired. In other words, we talked about last time the difference between a mutation occurring and a mutation being found. So it must not be repaired, and it also must be lucky, because if it occurs on a piece of the DNA and that DNA doesn't get packaged into a phage head, or if it does get packaged into a phage head and that phage never gets lucky enough to stick to another bacterial cell, then that mutation is lost. And finally, once it gets into the cell, another host in which it's going to grow, it must not be selected against. So there's a variety of things that are going against the mutation surviving, but there's also so many of them that a large number of them make their way through. So genomes do change over time. This is sort of repeat again of last week. DNA is in constant state of change. I talked about last time there are errors in replication, there's instability of DNA in solution, there's damage from UV light, chemicals, and there are repair systems that are there to try and correct the systems. We have small changes where we have a single nucleotide change, which can be any kind of one base to a different type of base. I didn't talk about the differences of being if you have a purine to a purine or a pyrimidine to a pyrimidine. That's referred to as a transition. And if you have a purine to a pyrimidine or a pyrimidine to a pyrimidine, then we have a transversion. So there's different types of mutations. We don't need to cover them this time. So we also have mutations that lead to an insertion or a deletion. So in this case, there's an extra base here. And if we just find these things in two different samples, we don't know whether this extra base arrived into a sequence or if the previous sequence that we had has a deletion of the base. And it has to be experiments then to figure that out. So the changes that we see, and I talked about this last time, in non-coding regions are more readily found, and it doesn't mean that they occur more readily. They're more readily found than changes in the coding regions, and this is a consequence that evolution of proteins, sequences, and the structures occurs more slowly than that of DNA sequence changes. The most rapid changes in the genome, particularly changes in protein structures, come, however, from horizontal gene transfer, and this is where you actually swap in sequences from someplace else. So we have changes that can be small changes or larger changes that occur over time, but a very rapid change can occur when you have horizontal gene transfer. The sequence changes may affect genotype and phenotype, and we've used these terms a little bit, but just to reiterate, a genotype is all or part of the genetic constitution of an individual or a group, and the phenotype is the observable properties of the organism that are produced by the change in the genotype as a response to its interaction with the environment. So one is what you actually have there, and the other is what do you see, and you don't always see when you have a change in the nucleotide sequence, and we talked about there being silent mutations last time. So that's the level at the small scale. This is three different phage genomes, and they're familiar with you. At least two of them are. So there's Shericon and TBR8012. The bottom one is ukulele, which is an E-cluster phage as well. It's the first one we isolated in the very first year of the phage course. And they're just lined up And you know now enough, you've seen this enough that you know the purple areas are areas where they're highly conserved at the nucleotide level and at the protein level, and the white areas are the things that indicate that there are changes. So in the purple areas, what are buried are the sequences such as I found for you in the study questions last week, where we have just small nucleotide changes. In this case, we have more significant changes, but again, they're fairly small, although I think this probably is involved with some sort of horizontal gene transfer, bringing in a lot of changes at one time. So we have this genome then, and we have these white areas that indicate that there have been some changes. There also, as I said, there's the areas that are conserved, and there's, the structural genes are conserved for a reason. Because the phage needs to know that it's got the right kind of protein to make a tail or tape measure. It has to have the right kind of proteins to make the head. It has to have active lysine A and lysine B in order to get out. So all those things are important to be conserved. There are other regions where things don't change, or where they do change a lot. Now, the conservation in some regions are dependent upon two things, one of which is conservation of the sequence for its function, and conservation of the sequence because it requires something else to function with it. So one example of this was that when we have, this is the box represents the integrase for these three phage. And what I've indicated by this arrow is the likely place for @P, and that's generally true, that the @P is located right next to the integrase. And there's one reason for that. The major reason for that is that integrases, of course, have some of their own unique characters. There's two major types of integrase. There's a tyrosine integrase and there's a serine integrase. And even within those categories, they have some differences in the sequence they recognize. So if you were to swap in just the integrase into the genome and not the associated @P, then that integrase has nothing on which to work, and so it won't work. You can imagine the same scenario occurring with if you swap in a new repressor protein, that if it doesn't have the right cis-acting elements on which it binds, if it doesn't have the right stoppers and operators to bind to, then it won't have any effect whatsoever. So you need to bring sort of coevolution, two things, the protein and the targets for those proteins. But there are regions where we do see wide changes, and one of the things that Hendricks points out in his paper, that in the lambdoid type phages, and the phages we work with are of that nature, you find two areas where you tend to see them, one of which is upstream, or rather downstream, or the space between where the lysine cassette is and where the integration cassette is. And so that's in this region. We see a large amount of variation. And the other end is towards the end of the right, the other place is towards the end of the right arm. So we see variation here, and I'm going to show you some striking examples where that's not always true, but that's generally true. That right arm tends to be much more variable than the left arm, and we have this little zone in the middle where there are changes. Why that's the case? I'm not sure if there's any experimental data that completely confirms it. Of course, it's right at the end. If this is the integration cassette, this is where it integrates into the genome, and so that's a place that might be vulnerable to mutations. And the same is true. This is at the end of a fragment or end of a genome, and so we have maybe some damage more to the DNA in this part. But that's speculation at this point, something that maybe you'd want to investigate. So just to give, this is just to zoom in more, carefully showing you what you have here. Is you can see from the white areas that there's not much level at the nucleotide level, and if you look at the colors and the boxes that are here, you see that they're different. So there's been a lot of changes here, and there's been a lot of changes here. We have other kinds of changes too, and Hendricks talks about this in his paper. So here's a case where Shericon, this is the terminase, this is the large subunit of the terminase, and here it is in ukulele, and here it is in TBR12, and it's bigger in Shericon. It has an extra sequence that goes from here to here. And that sequence is found in 49 of the 84 E cluster phage, but not all of them, but somewhere a little more than half, and 35 of them. So this is either insertion into 49 or it's a deletion from 35. And the thing about it is that whatever happened, it kept this protein perfectly intact in terms of reading frame from the start, which is here, And this start is the same as the start here and here in T-Rate 12 and ukulele. So it kept the reading frame right through this insertion or deletion and out the other side. And so one has to wonder, is there some mechanism during the event that makes sure it does that? And as Hendricks has pointed out in his paper, that's likely not to be the case. That if in fact we have an incorrect insertion or an incorrect deletion, that's very quickly going to get selected out. So this thing was not from nowhere. I mean, we can find sequences related to this in other phage. So this is the list of BLAST, the bottom part of the BLAST sequence, and down to this line, those are all E cluster phage. And then we get into other phage, and so little e is this is the predicted terminase for something from the cluster J. And as we get further away, we even find that it's being called something different. It's not even being called terminase anymore. But there is a sequence here that is found in other phage and somehow got itself in here, or it came out of here, and it has cousins someplace else. And we don't know at this point which way that goes. That's another thing that we might be interested in studying. So back to this image again, and I'm going to be finishing up fairly quickly. So this is the E cluster, and if I put on more E cluster, it would look very much the same. There's a very strong conservation in E cluster, except in these very few regions. But that's not true in some other clusters, and I'm going to give you two pretty significant examples, one of which is this, and this are the N cluster phage. And what you see in the N cluster phage is that here on the left arm, it's highly, highly conserved. There's almost no white out here at all. And a couple little spots in through here. And we see a little bit of conservation in this area as we get further and further out. But most of the right arm, in fact, half of the genome, has just been shattered and broken by things coming in that are just not related to each other. And so there's a huge amount of changes that have occurred in the right arm. There is some conservation as we walk our way down through here. As we walk our way down through here, where we're conserved at the place of the repressor protein, and we're conserved actually in this case at the very ends, but in between, it's a real mess. Now, that's in contrast. Here's another set of genomes. So this is the A2 clusters, and they have got some areas that are highly conserved, but the areas of mismatch are scattered throughout the entire length, and there's large numbers of areas of white. Now remember, clusters require 50% sequence identity, and they achieve it by having a 50% identity throughout its entire length, whereas in the case of the N cluster, they're really strongly conserved in one place, and the rest they don't have much sequence identity. So there's many ways in which genomes can evolve, and each cluster is going to be different in the way they do this evolution. And that's one of the things that makes it interesting, is how come this cluster does it this way, and the N cluster does it this way, and the E cluster does it a totally different way and hardly does it at all. And maybe it has to do with the repair enzymes that are associated with these particular systems, or maybe it has to do with something else. So in the end, phage genomes are wonderful tools to study evolution. They have single nucleotide changes, small insertions, deletions, large insertions, deletions. They have horizontal gene transfer. They evolve at a very high rate, and they have very large numbers. And all the processes that they use to evolve are the same ones that you will find in higher organisms. So what we learn about evolution from the phage can apply up the ladder to other types of organisms.